integrated protein and peptide search for the cell gene and its whole sequence. Dr. Tarro.
Thank you, Mr. Chairman, ladies and gentlemen. Good morning. What I'm going to talk with you will be a research that I started about 30 years ago. And so there is some Rembrandt lecture on anatomy without the section, because we go on dissection something. And we start with about 30 years ago papers in which we were able to isolate this complex of proteins from tumors, different tumors, and we tried to, of course, to understand what was the material, the stuff that we are dealing with. And one very important problem was, at that time was maybe more difficult, to sequence, identify what was at the beginning a big complex, 214 kilodaltons, and then 100 kilodaltons, when we were able to purify with the help of Dr. Marshak at Cold Spring Harbor Laboratories, with the blessing, I would say, of Watson and John Harlan. And so, I mean, we were able to obtain some semi-purified protein, some epitope of 8 amino acids, and then we prepared antibodies for this molecule. Of course, I mean, it was very important to get the rabbit cella, and then to show that it's very important to the reactivity not only with the original tumors, but also with the cell lines dealing with the same tumors. Another approach was done in Rome by Professor Garagi and his group. At that time he was working at the Tor Vergata University. Now, as you know, he's the director of Istituto Superiore of Sanità, Health Superior Institute in Rome. And he made a nice ELISA test just to look in cella if there was the possibility to have some marker for this protein. And at the same time, in Naples, at the time I was there, working at the Proteine Hospital, we were looking for antibody in the human cell from cancer patients reacting against the same protein. Then we were able to make some pathology studies concerning the histochemical analysis using polyclonal antibodies against the previous peptide of 8 amino acids, and then we will see shortly what we got. And at the same time, it was very important to see if there was in the activity normal or dysplasia or different kind of malignant tumors. Of course, I mean, it was possible to see the localization of this antigen, and then this was done in Rome, in Naples, and in Pittsburgh by Dr. Herbermann. So we can synthesize with some iconography from tumor cells. We have this antigen going to blood, and there are specific antibodies for this, and it's possible to measure by ELISA. And then we were able to show that in vitro it was possible to have hematogenic activity against this antigen. The same antigen was used in the light hypersensitivity test on the skin of people with cancer. And we can make some hypotheses concerning the possibility that this antigen was taken by the antigen presenting cell with